to have such a focus group on vaccinology. So yes, I will talk about the variation in vaccine responses, including, looking at the global south, which is not often studied, and linking them to the immune system. And I really like to emphasize when I give a talk how variable the distinction between what we know, variability also in our knowledge of the immune system. So if we're looking at textbooks, what we read is really the knowledge of the immune system as it is based on populations of affluent regions of the world, because that's where big immunological labs are based. That's where our knowledge comes from. And we know very little about what the immune system looks like in the global south, where the conditions are very different. And I think a lot of knowledge is gained from mouse, very important knowledge to be able to do real experiments. But what we also do is really study laboratory mouse, which is kept under very sterile conditions, relatively sterile conditions, and the immune system of that mouse is very different from the immune system of the mouse in the wild. I think what we do then with this knowledge of the immune system in terms of vaccine development is, of course, to use this knowledge that we know to develop vaccines, and that vaccines are tailored around the knowledge we have on the immune system of affluent regions of the world, the global north, and they could be used in global south. It's interesting that now evidence is accumulating over many years that some vaccines really perform less in less affluent regions of the world. So, we're doing all this vaccine development on the knowledge that we have, but, we don't realize that, this knowledge is restricted to a certain group of population and environments. And it might not be optimal for areas where people are exposed to different environmental factors. So I just want to really go through that with you. Basically, this is something that is being now reported, that, for example, if you look at the immunogenicity of the Ebola vaccine, you see that that's different. It's higher in the UK compared to Senegal, for example. If you look BCG, we know that there's a huge difference in how BCG is in its immunogenicity, but also its efficacy can be very varied. And immunogenicity in the UK measured by cellular responses can be high compared to when you measure it in Malawi, in Africa. Yellow fever vaccine is a very good vaccine, and there, it's not so much the efficacy maybe, but it's really the immunogenicity again that is different, higher in Switzerland than in Uganda. And the efficacy, which is really very important, for example, of the rotavirus vaccine, is high in high-income countries, but low in low-income countries. This is from large meta-analysis that has been conducted. And, that's in low-income countries is where the highest burden of death of newborns, infants is. And that's where the virus is needed most and is performing less. And, people will say, oh, it's to do with genetics maybe of the different population. Genetics is extremely important. But, when you go within a country, even within a country in global south, for example, you could see differences in immunogenicity of vaccines if you're in a more urbanized area compared to a more rural area. There are differences seen in immunogenicity of tetanus and influenza vaccine, for example, both antibody and cellular responses. So, there is definitely an issue there in terms of immunological responses. And for some vaccines, there's even efficacy seems to be different. I'll come to that towards the end again. And, what we have tried to do is to try and use high-dimensional immune cell profiling using CyTOF. We can use metal-conjugated antibodies, and, that can be really put through a nebulizer, which will then destroy the cell, but will release the metals that are bound to the cell. So you can identify the mass of the metals and therefore the antibodies that are on the cells. And that allows for many more antibodies to be used for analysis of profile of immune cells. And if we do this with flow cytometry, all the compensation is always a big problem. So we know 25 antibodies we can do nowadays with flow cytometry. You can do more, but that's when the challenge of compensation comes in. So using CyTOF, it's really been a way to analyze within one PBMC, peripheral blood mononuclear cells, to try and use the PBMCs and then use a panel of 40, let's say, antibodies and characterize the cells. So when we do that and look at what kind of cells we can identify profile within PBMC, just one vial, you can identify 100+ clusters from on the different cell lineages, CD4, CD8, monocytes, all the major lineages, and within those, the subsets and the clusters can be identified. And then you can really map those, project them into these profiles, and which you see here, and we can compare. These are healthy Europeans and healthy Gabonese living in Africa and Europeans living in Europe. And you really age and sex matched, and you look at the immune profile. These are adults. When you do that, you see that, for example, the really bright areas are where there is enrichment of, for example, a certain cluster types of cells in CD4 or CD8, etc. And what you immediately see, you don't even have to be an immunologist, just an artist can just see that there is a big difference between these profiles. And for example, here, a whole chunk of CD8 cells are absent in Europeans, present in Africans. If you compare gamma delta T cells or ILCs, a whole chunk missing is highly enriched in Africans. And so, the immune system, which we consider this is the type of cells we have and the characteristics of them, is very distinct between Europeans and Africans. This is in adults, but we've also started looking at early life. So these are cord blood cells from, again, Europeans and Africans. Again, Europeans are the Dutch, like the adults, and then Africans are the Gabonese cord blood cells. And when you do these studies, again, using mass cytometry, there again, you see a big difference in the profile of the cells very early in life. When we see these things, we are often saying, oh, it's all to do with genetics, as I said, very important. I'm not saying it's not, but if we go, sorry, yeah, this is so, just to show you that indeed very distinct profiles, particularly in NK cell, ILCs, B cells, and monocytes are seen. And in terms of, yes, it could be the countries, Europe versus Africa or Southeast Asia, where we do a lot of work. But, when you go within the same country, then you see also big differences. So here we have Kampala in Uganda, the capital city of Uganda, and then you go a few hours further, and there's a very different life living conditions, and exposures are very different. And here is Jakarta, a mega city, and if you go to the high, affluent people who live in Jakarta, these are the conditions they're living in. And then again, a few hours later, you go to an area where the living conditions are extremely distinct. So you can imagine that the exposures in the rural areas are very different, right? A lot of exposure to microorganisms and parasites can be challenging the immune system that are different from what the immune system here is challenged with. And, obviously nutrition also extremely different. What they eat is very different, and that can influence another factor that influences the immune system. And we go to these areas, we're interested in parasitic infections, have been for the last, few decades. And that's why, we go to these areas. I mean, we go and study parasitic infections in rural areas because that's where they're highly prevalent. Sometimes 100% of the population has these helminth infections. Malaria can be highly endemic. Many other parasites are present, particularly in rural areas. But as you go to more urbanized regions, we lose them. In the Netherlands, we hardly see them anymore. Whereas, 60 years ago in the Netherlands, there were also parasitic infections. So, the infection exposure is very different, as well as nutrition, as we realized. But if we then compare not only, Africa versus Europe, but also within urban and rural areas and compared to Europe, what we see is really a picture for Indonesia, for example. Here is our residents, age and sex matched Leiden people. It's all met with mass cytometry, looking at the differences. And what you see here, for example, Leiden and Jakarta, urban Indonesia, the profile look pretty similar. If you compare it to rural, rural really looks different. Look at these IL-10 producing B cells. They're enriched in rural Indonesia, but they're completely absent from both urban Indonesia and from Europe. So, the people, and also you look at the T cells, again, regulatory T cells and Th2 populations, which you can identify. They are present in the rural, but they're much less present in the urban, either urban Jakarta or the Netherlands. So this picture of living in rural area is very driven by the environment, it seems, that you live in. For example, these are just examples of cells that often come up when you compare rural urban areas: CD11C positive B cells, CD161 positive Th2 cells, and then CTLA4 and ICOS expressing Tregs. Tregs are similar, but when you dig into Treg, not only by doing FOXP3 and CD25, but really going into CTLA4 ICOS expression, then you start seeing differences between the rural urban populations. And it's not restricted to Southeast Asia or to, Indonesian rural urban differences, but rural urban differences are also seen within Senegal. That's where we have collaborations. Our collaborators work also in rural and urban areas, and then we compare them again with the Netherlands. And you can use the same kind of mass cytometry, same panel of antibodies, and you do see again that the three areas segregate very nicely. So Europeans, rural and urban Senegalese. And you can actually project that in a continuum. And the continuum really shows that the change in the immune profile is really going from the more rural to urban and then to the Netherlands. The colors are reflecting rural, urban, and the Netherlands. So there's really a gradient of change in the immune profile as you go from rural to urban and to Europe. And I just want to show you some examples of doing also a single-cell RNA-seq. What we see is that for example, you can find NK cells. Again, NK cells in rural area and the urban area of Senegal compared to Europe. And these are the pictures that you see. What you see is there is a whole cluster of cells present in rural, but very absent from urban areas. When you characterize them further, you look at the antibody that is bound to these cells. There's the program that is different, the transcriptomic, but also you see what is different is CD57 expression and, for example, TIGIT. This population expresses CD57 and TIGIT, and it's only present in rural areas. If you do functional of these cells, what you see is that, when you look at TLR7 and 8 ligand stimulation, that the NK cells from more rural areas are not responding in terms of gamma production, whereas they do respond in terms of gamma production when you stimulate them in urban areas. So functionally, it's not just the the profile of these cells that are different, but also functionally, when you stimulate them, you see very distinct responses. So this is, TLR stimulation, vaccine responses. NK cells are important, for example, in immunity against malaria, where you know that NK cells within the liver, if the malaria parasites are in the liver, we have a liver stage, we have the blood stage of malaria parasites. And what you see is, NK cells indeed, they have to produce their cytokines to be able to stimulate effector cells. They can also play a role in, antibody mediated cytotoxicity. And, so NK cells, certainly in immunity to malaria, are playing a role. We know that NK cells can also be important in vaccine responses. So here we are slowly trying to find, the variations in the immune system that we see in geographical areas that might underlie the poor responses to vaccines that are seen in some geographical areas. And I just want to go through the malaria vaccine. The malaria vaccine is being developed. There's one of them is the attenuated malaria parasite. And this is really when I think most of the differences in vaccine responses have been attributed to, the vaccine was not transported right. It was not injected rightly. It, within the comparison of how of the study designs can be very different. And therefore, you see discrepancy in vaccine results. But I think one of the most compelling data that shows there is a difference in vaccine responses comes really from malaria studies. And here, I want to show you this idea that, controlled human infection can take place. So what happens is that volunteers are asked to participate in studies, and they're actually infected with malaria parasites. Why? Because malaria you can really treat. If you have the treatment, you can stop the infection. So what you do, healthy volunteers, you give them malaria, and then you can stop the infection. You can see when the infection comes up. So let me show you. So basically what you do, you can use this story, and then you can use these models for testing vaccines. That's why they're developed, because very quickly you can see whether a vaccine works or not. Because you can vaccinate somebody, a healthy individual, right? You vaccinate them with your candidate vaccine, and then you infect them. You infect them with malaria parasites, for example. There are also infection models for other pathogens. A lot of you will know that. But for malaria, for example, you can give the sporozoites, which is the infective stage. You either can give it via bite of a mosquito, or you can have them in a needle and give people. So you vaccinated somebody, and then you see whether this vaccine is going to work. Because once you vaccinate and you challenge them, you can quickly see, are they infected or not? So when you give somebody malaria, you see that after a few days, the malaria parasites appear in blood. And if you vaccinate and then look at what happens to appearance of malaria parasites, you have somebody who's in green, is just not getting infected because it's protected by the vaccine. And in other people, the vaccine seems to improve their resistance because these malaria parasites come up, these red ones come up later than if you didn't have the vaccine. So the vaccine is delaying parasitemia. It's working to control the parasite or even inducing a sterile immunity. So very quickly, you can really see if a vaccine is working, and if it's not working, you stop. It's hopeless. But if it is working, you can also improve it, tweak it, or take it indeed to areas to test to endemic areas. It's a very effective, cost-effective way of testing vaccines. So this model was actually tested not only in global north, but also in global south. So basically not, in the one was in the US and Europe, people were vaccinated with this attenuated malaria vaccine. And then they were given a challenge. And in Equatorial Guinea, the same people living in areas where malaria is endemic, they were vaccinated, but they were also challenged with exactly the same procedure. And the same group of people were trained well. I mean, the procedure is very similar. And what you saw there, with a complete controlled way of looking at the efficacy of the vaccine, that there was a big difference. So here is really people are free of parasitemia after vaccination and challenge. Here in Europe and USA, almost everyone was protected. They did not develop parasitemia, whereas if you had the saline, so here you see in Equatorial Guinea, people develop parasitemia, or in Europe, but in Europe, you get parasitic. So, but then what happens is when you give them the vaccine, you see that people are just not behaving like in Europe or in the US. There is some improvement compared to the people who were receiving saline. Not as many people had malaria, but the difference between these two types of attenuated vaccines is really different from what you see in Europe and the USA. And that's what we mean. But in Europe and USA, you can get almost 100% protection. It's 100, and it depends how you calculate, 100 to 80%. But when you go to Africa, it's really, really much lower efficacy, and that's a problem. And this is the problem we're dealing with. And the efforts are really to understand which immune profiles can predict whether a vaccine is working well or not. And one of the ways we also extended not just to cytoph for ex vivo, looking at the phenotype, but also looking at metflow, metabolic enzymes, because the metabolic state of immune cells can also affect the reaction, immune reactivity. And also we've used, functional analysis of the cells. So we've stimulated cells with either PMI-N-myosin or with a TLR ligand, and then again done mass cytometry. We've also looked at IgG glycosylation. We've put all these databases together and using MOFA, to actually identify a signature. And in Senegal, we have identified a signature that distinguishes Europeans, as I said, Europeans from Senegalese, but also urban and rural Senegalese. And there are things that are distinguishing these groups. And if you take that and say, please predict from our Indonesian data set, so people, can you distinguish urban, rural Indonesians from each other as well as Europeans? This works. So you have a signature, and then the signature can actually distinguish Europeans from urban, rural Indonesians. So it's not just specific for Senegal, but seems to indicate that there is a rural signature that is working across populations. So basically what I want to say is that, if we can really dig into these signatures that are distinguishing populations, maybe we can understand how, influenza vaccine in semi-urban areas works better than in rural areas, or, in high and low income countries, rotavirus is, working better in high income countries versus low income countries. So, I mean, using really detailed analysis, using different omics procedure, maybe we can identify pathways that can indicate why you have lower responses, and then we try and change that. And this is really basically what we're saying. It's important to have a very broader view of the populations we study, not just global north, but also people living in global south and in different areas, different exposures. And then, compare low responders to vaccines with high responders to vaccines, so that you can identify the pathways that you can modulate and maybe supplement vaccines with these pathways to improve at the population level vaccine performance. I just want to take 2 minutes of your time, because you're a society dedicated to vaccine. We also have, and if you're interested, that we have a knowledge hub, which is called Hypovax Global. This is being created by some funds that became available. And basically, we bring people from different parts of the world. They come and train in Leiden. They come with their samples and they measure them using our facilities. And that means we can have information from different parts of the world. And this is really a network that works together. And you know, equity is very important in the data. They learn also to analyze data. So the data can be analyzed by everybody and not just the global north participants. And there's a lot of attention given to, you know, having the same rights in terms of, you know, if it ever leads to a product that everybody really benefits from that, the ones who are a member of this consortium. And their really goals are, you know, create a group that everybody works together, share best practices, and have metadata analysis together, centralized measurements. It doesn't have to be all in Leiden. It can be also in Uganda. There is a good facility for a certain assay. It can be done there. And basically, this is really something that we're trying to create together to fight the variation that we see in vaccine responses. And with that, I would really like to thank the study population and also different people who have been collaborating for many years and generating data, as well as our grant-giving bodies, ERC, Gates Foundation, the Dutch Spinoza, and NIH. And thank you for your attention.
Thanks a lot, Maria, for your presentation. It's like amazing how we differ in the response to vaccines that we mount. There were some questions through the chat that were asked during the presentation. And so I can start to ask them for the participants. The first question is regarding the exposure to malaria between people living in Africa and people living in Europe or US, because in Europe we are mostly naive. Maybe it's like one main variable between the population that you studied.
Yes, of course, you know, I think, you know, one of the issues is whether pre-exposure to the pathogen you're studying is already a big factor driving that. And that's definitely a possibility. And there's some evidence that if you treat malaria before, that you can actually help the vaccine to improve. It's still being debated. There's still, you know, people are, you know, some of them are saying, you know, that was not the way to set up the design, etc. But irrespective of that, I just want to draw the attention to the Ebola vaccine, because Ebola was not there in Senegal in the people that you vaccinate. Ebola was not, you know, in people in Europe, and still you see a difference in immunogenicity to Ebola. What I want to say is that that cannot be the only reason, because you also see differences in vaccine responses to pathogens where there is no pre-exposure.
Okay. Yeah, that's like a good evidence.
I mean, it's just one, but I'm sure, you know, it could still be playing a role.
Yeah.
And also, you know, you look at malaria, but and the same people have also helminth infections or not, or other infections, CMV. So yeah, I think the issue of is it really pathogen specific or not is important, because in terms of global health, you can, you know, try and get rid of the factor that is leading to this. But it has proven really very complicated. Also, helminth infections, you can give anti-helminth treatment, and it's been shown that it's not affecting it. So current infection is not. Maybe it is the wiring of the immune system, what you are interested in early life, that is now affecting responses, even though you have current infection. If you remove it, it doesn't work because it was the past exposure.
Yeah, your history.
Yeah.
Among the participants to the webinar, many wonder whether the microbiota or also gender might play a key role in the differences that you reported.
I think that's really important. There is, of course, really right now, I think one paper which has actually addressed that by antibiotic treatment to show that microbiome can have an effect, because, you know, we have to base everything on evidence, of course. So you need to remove, like the helminth you remove, some, you know, and the antibiotics, you try and change the microbiome. And that seems to have an effect. That's a publication by Bali Pulendra. It's a group. All I can say is that this is an area that I hope all young people will be working on, because I think it's an important thing to work on. And we are now looking in one study that we've conducted, looking at the vaccine response. We have collected some stool samples to look at the association of microbiome with the responses. I just want to say, I think microbiome doing in Global South can be very challenging. So, you know, collecting stool samples sometimes culturally is not as easy. Secondly, the temperatures are different, and how you collect it can be, so there are issues to deal with in terms of to get very robust data on the microbiome. That is not because you've used the stool samples you can't immediately sample. People don't like giving it immediately, maybe later. So there are these technical difficulties, but we've tried very hard to overcome them, to use it in this vaccine study. So yeah, we'll see. And I think other people are also. For sure, there are big differences in the microbiome between rural and urban areas in low middle income countries, between low middle income countries and high middle income countries. High income countries. So teasing that out would be really important and interesting for sure. Yeah. Thanks.
And did you have a chance to compare rural and cities within Europe or within the US, and whether you could document differences too?
You know, we haven't. And that's something that comes up all the time. And I think it would be really good if there are people who are working in rural and urban areas of France. We would be very interested, seriously, to look at that, because I think there are still in Europe, there are not that many, right? In the Netherlands, I think it's very difficult to find real rural places. You know, people say farmers are exposed to a lot, but the farmers, there are farms in the Netherlands that people don't even see the cows. Everything is so automated, right? I think in France, you still have Besançon. I remember people working in that area that have really traditional farming environments. I mean, it would be great if people are working on vaccine responses in those areas, we would love to work together. The other one is a possibility is also to look in Eastern Europe, because in Eastern Europe, you still have very rural lifestyles. So yeah, we are very interested in that. It's just how much can you do? I hope somebody will be interested and would like to hook up with us. Yeah.
Yeah. And maybe to come back, whether you could identify huge differences based on gender or based on
yeah, that's a very good point. Yeah. So you know, these immunological studies that we've done, when you do mass cytometry, you know that it's really low throughput, so it's impossible to do hundreds of people. It's always like 10, 15, 20 per group, making it a bit difficult. But now that we're trying to create this centralized database where you will get 100 plus data, then we're going to start looking at male-female differences. And, you know, it is known that innate responses are different. So I think we will probably start seeing things we have not done yet, because, you know, when we do it, it's only in small numbers, and then, you know, things are spurious. So I think we just need to enrich the data. Okay, I turned it on again. Did you hear me?
Yeah, now you're back.
Yeah, basically, we need a bit more time to enrich the data set a bit more, to have large enough numbers.
And there are also many questions regarding innate immune memory and training. So I guess it's not so easy to address and to take into account all of the variables that can impact immune responses to vaccines. What do you think about this past history of stimulation of the innate compartment?
Well, I think that's why I showed the NK cell in particular, because that's considered as an innate, and I mean, soon you will be hearing from fantastic work of Mihai Netea's group, who focuses on that. And I mean, that's one of the reasons I also showed the NK. You see clearly that the NK cells of the rural urban, they're different. And I think we have looked at their function. It's also different. So it'll be interesting to see indeed the level of at the epigenetics level, how they are. So this is one thing that we will be working on. I think I'm just waiting for, nowadays you have methods which really
You're mute again, Maria.
Okay, so I think it's really important to look. So I think now we know which cells of the innate cells might be interesting to target and look at the epigenetic modification of these cells. One of the questions I have generally is, are the 3-4 places that we look at, is that sufficient, or do we have to go more global? And like the cut and tag method that has been recently described, it'd be interesting to apply that. These samples are very precious. It's not always easy to get. So we sometimes also want to wait till we really are going to get the right the final answer when you use the one PBMC sample that is left.
What is left.
But I think this is the way to go, and I think this innate is really interesting. But yeah, next speaker is the world expert on that. So
yeah.
We are approaching it from first understanding which cells and which compartments, and also the transcriptome, and then to really go for we're gonna ask this question on that level. But you could also do it on a PBMC level and see what is different. Yeah.
And do you have a chance to get access to samples sampled very early post immunization? Because usually the way clinical trials are designed, of course, they are focused on late time point when you get antigen-specific responses.
Yeah, so that's why we have this cord blood study. It's of course not after vaccination. It's very difficult to have blood in volume sufficient to do these studies, but to be honest, now there are these ways of preserving whole blood. So even a drop of blood could give you a little bit of cells that maybe you can pool, and then from different babies, and then look at pre and post vaccination. We don't have. We do have from cells that we will have access to, and it's planned to be studied not after 1 year, 1 to 5 years of age. It's still not very early, but it's not, yeah. So I think technologically it's going to become possible to do that slowly, and whether it's us or other people, as long as it gets done, will be very nice. Yeah.